it's a great pleasure. Well, okay. So it's a great pleasure to introduce and to have visit today Dr. Gea-Ny Tseng. Gea-Ny is one of these people who I don't know how often we get to interact, but I just feel very close to, maybe for many reasons, certainly included in them, like-minded interest in channel biology and a particular focus on channel trafficking. And can you hear me? Okay. Okay. So especially when it comes to trafficking, I need to shout. All right. And so I feel like in many ways we're kindred spirits. And so Dr. Tseng started out at National Taiwan University, which is, I don't know if you know, but the same institution as my hero and mentor, Lily Jan. Yeah, Lily and Yunnan, actually, came from National Taiwan University. And for those, that's saying something, okay? And then she got her master's and her PhD with Columbia University. This is the home of much ion channel in the sort of latter part of the last century. And stayed on at Columbia University until 2005, was it? Stayed on the faculty, 2000, excuse me, 1999, 2000. Then moved to Virginia Commonwealth, right? I advanced to professor in 2005, and it's been in Virginia Commonwealth ever since. Dr. Tseng's many discoveries include a focus on palmatolation, which in the simple-minded way of thinking about it, and that's as far as I can go, is adding a palmatoid group to its cysteine residues, right? Yeah, and in doing so, by affecting these residues, that considerably changes the mobility, the trafficking, the membrane affinity of proteins, especially membrane proteins. And so I think it's very safe to say that she is the national expert on this event when it comes to cardiac, certainly when it comes to cardiac ion channels, and then in general, the effect of trafficking membrane proteins and receptors such as adjunctive in association with the dyad. And so Gea-Ny's really been a strong force nationally, both at NIH as well as the recent FASA meeting. It was a couple of years you did that, right?
We did twice.
Twice, yeah. And so really it's, has sort of set the trend of how we as ion channel biologists need to drive forward. So it's for these reasons, we're here, invited Dr. Tseng to basically learn from her, okay? And so with that, the title of the talk is Traffic Control of IKS Channel in Ventricular Cardiomyocytes. And welcome to Salt Lake City and the CVRTI.
Thank you. It is a great honor to have a chance to present our work here in this institute where landmark studies of research breakthrough have been accomplished over the last half century. It's really honor. So the story I'm going to tell you, the traffic control of the IKS or slow delayed rectifier channel in ventricular myocytes began here 26 years ago. So this is a screenshot taken from the website, citing the long QT work by Sanguinetti and Mark Keating and Dana. I would like to add two more landmark papers. First one is Nature Genetics, reported by the Mark Keating and his group, describing positional chromium of a novel potassium channel gene, CVRTI. And in the same year, again, in Nature, with Serenity, Keating, and others, describing that. I'm sorry.
Just one. Would you be able to stand here? Otherwise, the camera's getting the back of your
okay.
head.
Oh,
If you
okay.
stand here, then they
Okay,
can see you.
Yeah, okay, then let me try to, okay, I can still, okay,
Okay,
sure,
thank you.
no problem, sir, thank you. Reporting that co-assembly of KVRQT1 and the small protein called MINK or ISK can form the slow delayed rectifier channel. So these studies are important for many reasons. One of them is that they ended the 9-year debate as to whether the small MINK protein, 129 amino acids, one transmit domain by itself can form the slow delayed rectifier channel. And second is that it identified the molecular basis for long QT1. So this was taken from one of their figures using a mammalian CHO cell line for expression, expressing MINK alone, no current, KVRQT1 alone, very small current, not that impressive. Putting these together gives you this beautiful, slowly activating our current, similar to the IKS current that one can record from ventricular myocytes, in this case from a dog heart. So MINK and KVRQT1 have several names. I will call them as KCNE1 or E1, KCNQ1 or Q1, and Q1 plus E1 for IKS. So, so these studies open a research field because people are interested in how MINK can modulate Q1 to make this very unique gating phenotype. So a search of PubMed 3 days ago using a string of keywords like KVRQT1, KCNQ1 retrieved 9,117 papers. A highlight of this research came across in 2000, it occurred in 17 and 20, where Rob McKinnon's group reported the 3D structures of Q1 associated with comodulin, a calcium sensor in the cytoplasmic domain, without or with KCNE3, a close cousin of KCNE1.
In the 17-1, I'm just curious.
So now the function of IKS is a major component of so-called repolarization reserve in ventricular myocytes. The best example data to illustrate this point is shown here using guinea pig ventricular myocytes under basal conditions. IK1, the inward rectifier channel, IKR, the rapid delayed rectifier channel, provide enough repolarizing force to repolarize a ventral action potential. IKS is very small. However, with beta adrenal stimulation, when the heart rate is fast and the L-type current is large to increase cardiac output, we need extra current provided by the IKS to shorten the action potential duration. Therefore, the heart will have enough diastolic interval for chamber filling with fast heart rate. And therefore, this explains why LQT1 patient and likely LQT5 patients suffer the highest risk of arrhythmia during exercise when the beta adrenal tone is high. On the other hand, we need to keep IKS small and slow in activation. If there's too much IKS, as seen in some KCNQ1 genetic variants, that can lead to short QT syndrome too, with risk for atrial fibrillation. So with this background, I'm ready to ask or present data for 3 questions, major questions I will with this presentation. First, what controls the IKS channel current amplitude in ventricular myocyte? Specifically, where and when Q1 and E1 assemble to form the IKS channel? It's a valid question. It has been actually debated for many years because Q1 and E1 need to come together to give the function of IKS. They are so-called obligate partners in cardiac myocytes. Then, do they always stay together? If not, where and when do they assemble? So next slide would talk about the history. Some people believe that Q1E1 assemble early, maybe translationally when they are in the ER, but opinion varies between whether Q1 is, sorry about that. Q1 is required to chaperone E1 from ER to plasma membrane, or E1 chaperones Q1 to the plasma membrane. Some people believe that they assemble not in the ER, but in the pre-Golgi domain. And some people believe, no, it's the assembly happens at the Golgi. And some people say, no, it's at the junction between ER and membrane. So the thing that the field is confusing, chaotic, just like this slide. So it is important to point out that these studies were conducted in expression systems, including oocytes. And we need to carry out the study in cardiomyocytes, more specifically adult ventricular myocytes, where IKS fulfill its physiological function. So this is begin data from my lab. We use canine ventricular myocytes. Spoil my surprises. Okay, so canine ventricular myocytes express IKS. We use antibodies to monitor the distribution pattern of native Q1, native E1 simultaneously in myocytes. You see this very strong striation pattern with Q1 and E1 cluster to the surface. They don't seem to stay together. There is some sporadic colocalization on the lateral surface, as shown by this cross-sectional view. So one question, easy question, could this be an artifact because of myocyte isolation, that protein moves around? And no, because if we use the same antibodies to stem canine ventricular myocardium, we see striation with Q1, lateral surface with E1. Could this be an artifact? Non-specific antibody bindings. So we answer this question by using GIP tag Q1, this red labeled E1, using adenovirus to express them in myocyte and see where they are. So here is the answer. Without any antibodies, just incubating the cells with these adenoviruses, where Q1 installation, E1 cluster to the cell surface. So, sorry. Okay, so they are largely segregated in myocytes. They seem to colocalize sporadically on the lateral surface. Now, localization close to the surface does not necessarily mean they are on the surface. So our next question is, are they really on the surface? On the surface, on the cell. Instead, then we use a Q1 antibody that target the extracellular S1 loop of Q1. We engineer an hemagglutinin epitope to the extra domain of E1, so we can use HA antibody to label cell surface protein. Now, without membrane permeabilization, meaning intact cell membrane, Q1 is an antibody in this sporadic labeling on the surface, whereas the GIP fluorescence signal representing the total protein has this very strong striation and perinuclear staining, reflecting protein translation in the nuclear envelope. If you permeabilize the cell membrane, apply this antibody, then you have the striation and perinuclear staining. Now with E1, without permeabilization, HA antibody stain is very nice surface. And total protein has these particles as well as some striation reflecting protein translation at the junction that's permeabilization make the two well colocalized. So they are colocalized on the surface of the myocyte, not inside or PQB. Now, colocalization may not mean assembly into functional channel. So we use proximity ligation assay to address this question. Are they really close enough to form a channel complex? To do that, we, sorry, spoil my surprise again. So let's go back. Okay, no surprise. So primary antibody, a rabbit antibody targeting this rat, a goat antibody targeting GFP, secondary antibody targeting this primary antibody from different hosts with nucleotide, with complementary sequences. When the two nucleotides are close within 40 nanometers from each other, they allow an amplification reaction, incorporating fluorophores, generating the PLA signal. So, in a rat mouse site expressing Q1 GIP, HAE1 this rat, the PLA signal, the puncta, are clustered around the cell periphery. So they assemble into functional chain of there. Okay, no problem there. Now, with this background then, how do they separate the mouse sites? Could they be translated in different locations? Meaning Q1 translated junctional SR at diets, and E1 translated by junctional SR just beneath the surface, creating this segregation. So to answer this question, we track the messenger RNAs using fluorescence in situ hybridization. We also monitor the protein using this color scheme. So in a red mouse site expressing Q1 GIP, the protein has this striation and perinuclear staining. Now let's wait for the messenger RNA. Yes, messenger RNA particles cluster around nuclei, more so in one than the other. And the combination of the two just show what they are not quite the same. With another E1 red ventral myocytes expressing E1 this red, the protein has this very strong puncta or vesicular compartment, and with cell ends or intercalary disc staining. Let's wait for the messenger RNA particles. They are also clustering to the nuclei, more so with one nuclei than the other. So this data suggests that Q1 and E1 are translated mainly in the rough ER compartment in perinuclear zone, or more specifically in nuclear envelope. And they seem to traffic to different destinations. Yes.
Sorry, I hope you don't mind that.
Please.
That's a gorgeous colocalization. Does that mean there's not local translation? You know, is it a central translation or is it?
That's why I use the word mainly. I think it's in the adenovirus expression is mainly in the around the nuclear envelope.
At the very least, it's mainly.
Right. Yeah. But in native state, I think junctional SR has the ability to translate membrane proteins.
Junctional SR.
Right. Junctional SR. Yeah, well, I have one slide to point in that direction, but I think Tsai at Ohio State University published a paper with another group, of course, showing that messenger RNA, actually translating messenger RNA is actually distributed in the myocytes away from nuclei and very close to the intercalary disc area. So, but I think it's an open question. I mean, how the messenger RNA know where to go to make the translation.
Neurons, there's no question.
Yes.
It can be distributed.
Right.
So the same maybe, but at least the majority is local.
With the adenovirus expression. Yeah. Let me point that out again. Right. So it's overexpression, adenovirus.
Yeah, I've always wondered, I don't know what the. This is great data.
We try to titrate the dosage of adenovirus. So when we do different experiments with comparing native with adenovirus express, we probably have a 50%, but less than 100%, or less than double the amount of native alone. Yeah. That's all I can say. Yes. Okay. So different pathways of traveling. So channels can traffic by different pathways. This is so-called conventional trafficking pathway, where channel is translating rough ER, goes to Golgi and plasma membrane in vesicles. But there can be unconventional pathways, and there are several possibilities. Channel may go from ER to the plasma membrane, may detour through this endosomal compartment, or they can go through Golgi, but in vesicles different from the convention, those in the conventional pathway. So we incubate canine ventricular myocytes with adenovirus viruses carrying Q1 GIP and E1B thread for different durations. At this time, specific times, we fix the myocytes and see where the proteins are. So I will show you X, Y, and YZ plan, but it's the major point is the time. So 10 hours, they are mainly around nuclei, consist with major translation sites. 14 hours, 18 hours, 24 hours, you see that the Q1 is spreading outward from the nuclear envelope in striations, finally reaching cell surface. On the other hand, E1 is in this very prominent vesicular compartment and eventually reaching the cell surface. At 36 hours is more similar to the native protein now. Disappearing from the perinuclear zone, disappearing from the vesicles, focus still in striation and on the lateral surface. So now to understand this better, we need to know this gets to your question now. The distribution of organelles involved in conventional secretory pathway. So this has been done with Core7 cells. For example, connexin and BIP, a membrane and luminal protein in the ER involved in protein folding, label the ER for protein translation. Vesicles going from ER to Golgi can be labeled by ERGIC 53. Beta-cope and Huizhang agglutinin will label Golgi, trans-Golgi, and post-Golgi, and then plasma membrane. But what are the patterns in myocytes? So, connexin and BIP label junction SR in the striation pattern. Also, nuclear envelope, more so with BIP than connexin. ERGIC label a very prominent vesicular compartment in the cytosol. Beta-cope and WGA has different patterns at different locations. I will point out this very prominent granular structure. So it's probably like neurons with long dendrite or skeletal muscle, where Golgi is not centralized in a location with a big structure close to nuclei, but is distributed into mini Golgi, arranged mostly longitudinally in myocytes. With that, then let's look at this E1. The vesicles colocalized with WGA and beta-cope. With Q1 GIP striation, really not much vesicle at all. So we concluded that E1 follows conventional secretory pathway, but Q1 does not. To understand this better, we use a strategy called retention using selective hook or rush. So with this procedure, we need to first have rush plasmid, where you have the plasmid can encode two proteins separately with this internal ribosome entry site. We also need to have stratavidin core domain and stratavidin binding peptide fused to the ER hook protein and the protein of interest. The ER hook protein is a protein with a very strong ER retention signal, so it stays in the ER. And protein of interest can be anything that you want to follow. So when this rush plasmid is expressed in cells, the protein of interest, spoil my surprise again. So the protein of interest anchored at ER by bonding of the two conjugates by the ER hook. Adding biotin to the medium, biotin will diffuse to the cell membrane, bonding to the red protein, releasing the protein of interest. Therefore, the protein can leave the ER, trafficking its secretory pathway. And so our experiments was done in Core7 cells. And so in the first set experiment, we have ER hooked HAE1 dysregulated co-expressed with another protein, VSVG, also ER hooked, labeled with GIP. So VSVG, I think it's called vesicular stomatitis viral protein G. It's a model protein that can travel from ER to the plasma membrane constitutively. So it's a good model protein that people use to track protein secretory pathway. So before biotin, both are in the ER. Half an hour, they start to leave in ER into small vesicles, coming to Golgi close to the nuclei. 60 minutes, larger vesicles, some reaching the cell surface. 2 hours, largely disappearing from the Golgi, not in the ER, in vesicles, and on the cell surface. Now, with ER hooked Q1 GIP, we co-express it with an ER marker labeled with this red. So we always have know where the ER is. So before biotin, half an hour, 1 hour, 2 hours, Q1 GIP largely stays in the ER, co-localizing with the ER marker. So then we've concluded that ER follows the conventional pathway, reaches the plasma membrane constitutively. On the other hand, Q1 appears to follow an unconventional pathway. But which one? There are many different possibilities. So to answer this question, first I need to clarify an issue. What is the nature of Q1 striation in ventricular myocytes? Essentially, we really cannot tell by light microscopy. The striation can be T-tubule or junctional SR. The two membranes are very close to each other. So I'm going to present three sets of data. Hope that I can convince you that there's a major component of Q1 in junction SR. So first, in myocytes without T-tubules, atrial myocytes or detubulated ventricular myocytes, shown by this WJ staining, no striation. The native Q1 is in very prominent striations. On the other hand, the Q1 striation overlaps with ER-SR markers, for example, RIR2, jungle feeding 2, and others. No problem. We can express this Q1 GIP and U1 dysregulated in myocytes, and we immunoprecipitate with GIP nanobody in this case, and dysregulated rabbit antibody, and we look what are present in immunoprecipitate. The upper part simply show that these antibodies co-IP IP very well, and the two proteins co-IP with each other. This is important. These SRER proteins, RIR2, SERCA2A, calsequestrin 2, calnexin, jungle feeding 2, they are abundant in the IP land, but much weaker or not detectable in the E1 dysregulated IP land, suggesting that Q1 is in an ERSR enriched environment, but E1 is not. But the best experiment is shown next. The third experiment, where we try to separate the SL, the sarcolemma vesicles, from the SR, sarcoplasmic vesicles. We run Western blot to try to quantify Q1 and E1 in these two membrane environments. So this was done with my good friend Ming Zhang, a summer visit from China, and we decided to follow this protocol published in 1989. So we homogenize guinea pig hearts, creating heterogeneous vesicles. Then we incubate the vesicles with calcium oxalate and ATP at 36 Celsius. So under these conditions, vesicles with calcium pump activity can pick up calcium oxalate and become heavier. Because SR vesicles have higher calcium pump activity in SL vesicles, they become heavier. And so we can then use ultracentrifugation on sucrose gradient to separate the two. So the tricky part is the incubation. It took us really the whole summer to try to optimize incubation. And the data shown here, the photo was taken the night before Ming's departure back to China. And you see that dirty fingernail, that was me. I was so excited, I don't care. So here, with 5 minutes of incubation, we could see clear interface between the sucrose gradient. So I then solubilize this fraction and run Western blot. So you see that SRER markers, ranolidine, sarcotocalnexin, are more abundant in the heavier fraction enriched with SR vesicles, suggesting they are enriched with SR. Now, sarcolemma markers are glycosylated sodium calcium entranger, sodium pump alpha subunit, more abundant in the light or SL enriched fraction. Q1 more abundant in the heavy SR fraction, and E1 more in the SL fractions. So no myocyte isolation, no virus expression, fresh guinea pig hearts. This is what we saw. Oh, this is data summary. Okay, no problem. So now this will suggest that Q1 sitting in the junction SR, very close to the plasma membrane, can maybe directly exit ER to the plasma membrane. This cannot happen as a free Q1, must be happening in the vascular form. So can we find Q1 vesicles moving from ER to the plasma membrane? So we began our hunting for Q1 vesicles going from ER to the plasma membrane. We tried different protocols. One is to done in those rush experiment, 60 minutes after Biotin application, some of the Q1 vesicles seem to be coming out of ER in these very small, tiny green vesicles that can be detected by the structure illumination. With live COS7 imaging, again, co-expressing Q1 GIP and this red ER, we sometimes seem to detect green structure budding from the ER and even have some indication that is moving toward the cell periphery, the plasma membrane. The best example is shown with this experiment where we used turf and wide field to monitor fluorescence close to the plasma membrane and inside the cell. And Dr. Berners, my collaborator, split the camera. So the image between these two is separated by 15 microseconds, almost simultaneously. So again, we co-express Q1 GIP and this red ER. So we have Q1 vesicles close to cell membrane, ER network deep inside the cell. And I point to several vesicles that I want to bring your attention to, especially this one here. This one seems to be pulling away from the ER, creating this tubular structure. Second frame, 0.65 seconds later, that little one becomes stronger, suggesting it's moving toward the plasma membrane. Another frame is gone. So our interpretation is that little vesicles is kind of peeling off from the ER, moving toward the cell membrane, fuse with the cell membrane, and disappear. So, I think we are very happy to see this. So we think this Q1 can directly exit ER to the plasma in vesicles. However, this process does not happen consecutively. So what is the cue for Q1 to exit the ER? Back in 1990, long time ago, it was described by a Japanese person, one person, that if the calcium concentration in the pipette solution is elevated by this pipette solution exchange device, there is an increase of IKS in guinea pig ventricular myocytes. We know that Q1 has this comodulin, the calcium sensor, constitutively bound to its C terminus. Now, two papers published in 2006, I'm using data from one of them, they report similar findings. In oocyte expression, Q1 plus E1 or Q1 alone will become smaller after injecting beta into the oocytes to reduce cytoplasmic calcium. Furthermore, co-expressing Q1 with this comodulin mutant that cannot bind calcium create this very unique channel kinetics, suggesting that is not calcium, comodulin is not binding calcium and creating this. So this data suggests that calcium binding to that calcium sensor comodulin can promote Q1 to the cell surface. How about mammalian cell? So this is our experiment. We co-express AT1 receptor with Q1 and GIP and E1 dysreg in COS7 cells. Then we incubate the cells with angiotensin to 1 micromolar for 1 hour before patch clamp or bioaccumulation experiment. The idea is that if there is a G protein PLC activation, IPV production, IPV receptor activation, releasing calcium to the ER, there could be an increase in surface Q1. And indeed, patch clamp of Q1 alone, Q1 plus E1, angiotensin to pre-incubation increase the carbon. This is data summary. And bioaccumulation experiment, co-cell lysate versus bioaccumulative fraction. Angiotensin to incubation increase cell surface Q1, but not E1. If we express comodulin mutant that cannot bind calcium, so might spoil my surprise. So we don't have the increase in current, we have very little Q1 on the cell surface. Whereas if we put 2-APB, a blocker of IPV receptor 5 micromolar during the angiotensin to incubation, we don't have that current increase and we don't have the increase in surface Q1. So all this will bring me to the answer for my first question. We believe that in myocytes, newly translated Q1 and E1 traffic by separate routes to different destinations, Q1 in junction SR, Q1 on lateral surface, and they co-assemble into IKS sporadically from Q1 reach the cell membrane. And just to have one more thing, this is the same thing actually. So the Q1 is at this strategic position in the junction SR with this cytoplasmic comodulin facing the didactic space. So each time you have a contraction, easy coupling, you have a little dose of calcium bound to the comodulin that will allow Q1 access to the plasma membrane. So this could be a good mechanism to keep IKS small, can be keep it in place with the calcium influx and preventing shock QT and lower risk for atrial fibrillation. So I threw in this figure last night just to show that Q1, E1, when they are on the surface, they have no difficulty finding each other. So here we have COS7 expressing E1, homogenize to create E1 vesicles, inject into oocytes, wait for FIORs, and do our 2 electro voltage current. And without injection, oocytes is a very small current representing native E1 at Q1. Several hours, 3 hours in this case, after E1 vesicle injection, we start to see this nice, not so nice, it's very small, but slowly activating IPS current, similar to what you will see in a Q1 oocyte injecting both Q1 and E1 RNA. So this brings me to my second question. How does Q1 trafficking play a role in beta enhancement? That, of course, is very important because that is a physiological function of IPS in the heart. So beta adrenergic stimuli induced increased IPS occur by several mechanisms. The first one is a speaking mechanism because the IPS deactivation is very slow. So if a fast heart rate, there will be a summation of AKS activation give you much more outward current in the early plateau phase, give you shortening of action potential duration. Another one is the PK mediated phosphorylation of casing Q1 channel components, Q1 and UTL that anchoring protein. But there's a third mechanism, a trafficking mechanism, that you require intact microtubules for AKS to respond to beta adrenal stimulation. So this was shown by a paper published in 2008 using guinea pig ventral mouse as a model system. Applying memory permeable CK-AMP to the guinea pig mouse cells give you a huge increase in IKS. If the cells have been incubated with colchicine to depolymerize microtubules, then CK-AMP gives you much smaller increase. And this is data summary. So back a few years ago, we tried to find Q1, E1 interacting proteins in cells by doing proteomics. So we express this in cells, immunoprecipitate, and send the samples for proteomics. So, and this is the table published in that paper. I want to bring to your attention to the finding of EB1, connexin 1 in unique IP from Q1 GIP, meaning trafficking on microtubules. So EB1 is microtubule plus and binding protein function as dimers, important for microtubule dynamics and important for trafficking of connexin 43, as shown by Dr. Shaw, and as well as sodium channel to intercalate disc of cardiomyocytes. So, but what about Q1 and E1? So I'm going to show you some data from red ventricular myocytes transduced with Q1 GIP and HAE1 dysreg. First set of experiment is to immunoprecipitate this protein and see what we can find. So we use GIP rabbit antibody to IP Q1 GIP HAE1 to IP HAE1 dysreg, and we also use E1 red antibody to IP native EB1 from these myocytes. Then also we have whole cell lysis as party control, negative party control without antibody to show the specificity of the IP. And we monitor this with GIP Go antibody, this rabbit antibody, and EB1 red antibody. So the first set of experiment, the point is here. Native EB1 can co-IP Q1 GIP, shown here. And QR, of course, is IP by itself, and HAE1 can co-IP as well. Now, the point here is that E1, native EB1 does not co-IP HAE1 dysreg because it's not here by dysreg antibody. Here, with EB1, the point is Q1 GIP co-IP EB1 as dimer. HAE1 dysreg does not do so. So it's a specific Q1 and EB1 interaction. Now, how about their distribution in myocytes? So we use GIP fluorescence protein to detect Q1, using that external antibody to detect surface Q1 before membrane permeabilization. And we use the EB1 red antibody to detect native EB1 after membrane permeabilization. And can we focus on cell center? Surface Q1 and EB1 seem to colocalize at cell ends close to ICV. We look at the cell surface. We see that the surface Q1 and from puncta colocalized with the puncta of native EB1 right beneath the cell surface. So we suggest that EB1 may be the guide for Q1 to exit junction SR, reaching the cell surface in myocytes. So coming back to this question number 2, I would suggest that beta adrenal stimulation increase the L-type calcium current, increase the calcium store in the ER, therefore increase the diabetic calcium concentration do easy coupling. That will provide more encouragement for Q1 to acid junction SR to the cell membrane. Yeah, now let's come to the third, and I think the timing is all right. We have time for a question. So how is Q1 E1 trafficking altered in disease heart? And what the functional consequence? I will use 3 examples. 2 of them have been published, and I thought I have everything. Every question answered. There come the third one. It was very recent, but very consistent, reproducible in 3 independent experiments, so I cannot ignore it. So I will show the data. So, okay. In old, about 2 years old, spontaneous hypertensive rat, we have hypertrophy, fibrosis, thrombosis in the left atria, suggesting atrial fibrillation, not happy heart. When we isolate myocytes from the young, apparently healthy and old SHR heart, and we use HMR1586, a specific Q1 blocker, to separate IKS from all the other ITO and whatever. We see that the IKS seem to become larger in the myocytes from old, unhappy, unhealthy SHR heart. With this immunoprecipitation, this is summary, there was maybe a little increase in Q1, but really not that significant drop of casein U1 protein. So this immunoblock total protein level does not explain increase in IKS. So confocal. Q1 in young SHR myocytes has this strong striation, but translocate to the lateral surface, well colocalized with E1 on the surface. And I think that we believe this may be the major mechanism for increase in IKS. Another example is a canine model of chronic premature ventricular contraction, PVC. This was created by my former collaborator, Dr. Huizar, and this was from his 2011 paper with by Gemini. Every extra systole induced by pacing with 250 milliseconds from the QRS for weeks would cause a gradual decline in LV ejection fraction. We collaborate with Dr. Huizar to study what happens to the action potential, what happened to the channel, protein distribution, and whatnot. What we found was that with IKS, Q1 seemed in striation, but seemed to be reduced in striation and moved to the cell end, and E1 is downregulated. Hash clamp using this protocol so that we can claim that pair curve being specific IKS is increased. Doing the SLSR fractionation, we see that Q1 is in the heavy SR enriched fraction in control, but much more abundant in the light SL enriched fraction, consistent with surface expression. So all is good. And I thought I answered all the questions. Here comes the surprise. Okay. Let's not do this. Let's come to this. So over the years, we see that Q1 is moving around depending on the condition. E1 seems to be very stable, sitting on the myocyte cell surface. And this is surprising actually, because as been shown by Jeff Abbott, that mean K or KCE1 has endocytosis motif in the cytoplasmic domain, and expression in either O's or expression or mammary cell, E1 tend to disappear from the cell surface with time. So there is inconsistent finding, inconsistent with our finding. So one possibility is that E1 may be binding to cardiac selective cytoskeletons. For example, TCAP, the cap protein capping tetin in sarcomere, and 4.5 link domain connected protein to FHL2, both have been shown to bind casein E1. But these are sarcomere proteins. They don't they are not consistent with lateral surface localization. Another possibility, back to my interest in permutation, we have some data suggesting that KCNE1 with its single cysteine residue in the cytoplasmic domain may be permutatable in mouse. Permutation will enhance protein anchored in lipid raft domain, and lipid raft domain is enriched on the lateral surface in mouse. So, with my collaboration with Isabel, we study how chronic PKA or persistent PKA activation for 12 hours in myocytes may influence EB1 and sodium channel and whatnot. So, the byproducts Q1 again, even this one. So we see that with PKA activation for 12 hours, there is an increase in cell surface Q1, either with Q1 alone or Q1 plus E1. Good. So it's considered with EB1 organization. Control, very nice surface signal. 12 hours of PKA gone. So, and either E1 alone or E1 plus Q1. So this is consistent with their separate trafficking. So again, 3 independent experiments, reproducible. So I think what is happening may be that E1 may appear to be very stable on the cell surface. It's actually going through dynamic endocytosis and recycling in myocytes. And therefore, it's not static, it's a dynamic presence. And therefore, I need to check that. Then I need to, if that is true, I need to understand how this endocytosis and recycling are regulated, and why PKA persistent activation will cause E1 to disappear from the cell surface. Maybe endocytosis, but not recycled. And with this increase in Q1, but decrease in E1, what happens to the IKS channel function? So, acknowledgement, funding, no problem. And I had no problem, meaning we have to have funding, otherwise we're in trouble, right? No, I struggle like everybody else. Okay. I have the pleasure of working with students and Sam and Akshay and Maddie and Zach, medical school now, Easy School of Pharmacy, Suklin Dental School. Will and John are probably still waiting for medical school admission. I have the pleasure of collaborating with Minjan, John Ho, proteomics, and Isabel Kishan, Ohio State University. Now with notice of award just yesterday. We know now we have the protein permutation grant beginning today. Official day. Now I'm going to
start here.
Right. Yeah, especially just 13 and 13 Tuesday, notice of award. Official day 15 today. So I'm going to collaborate with Trixie, proteomics, NYU, Jinzi, biostatistics, genomics, and Titus microscopy. So this I'll be working on for the next 4 years, maybe 5. We don't know. Okay, good. So I'm done.